Hello everyone, welcome back to our channel. This is Science Lectures 256. If you haven't yet subscribed, liked and followed, please do so. So last time we looked at membrane boundary receptors and we said there are of three types. Number one, we have what they call the G-coupled protein receptors. The G-coupled protein receptors. Then we said they are what they call enzyme coupled receptors. Enzyme coupled receptors, and lastly we have what they call ion gated channels. Ion gated channels. And in our previous videos we discussed about the G coupled protein receptors. So today we are going to look at the enzyme coupled receptors. So this is a special type of receptors that has an enzyme on its cytoplasmic tail and a receptor on the cell surface membrane. And there are of three types. Number one, we have what they call receptor tyrosine kinase receptor. Enzyme coupled receptors. We have what they call the tyrosine associated receptors, and we have what they call the theonine serine receptors. The theonine serine receptors. And I want us to begin with the receptor tyrosine kinase. Tyrosine kinase. So if I'm to try to sketch the ultrastructure, it would look like this. This is my cell membrane. I'm assuming this is my cell membrane. So on its surface it has a receptor, then it has an alpha helical structure, then it also has tyrosine proteins on its intracellular domain or side. So it has a receptor here, then there is this part that spans across the cell membrane, and you have the tyrosine this side, inside the cell membrane. And this receptor tyrosine kinase usually exists as one within the cell membrane, what we call monomers. Mono means one, so they usually exist as one. However, these receptors are very special, that whenever the signaling molecule binds, the dimerize. Di means two. So they work together as a pair, but they exist as one, what we call monomers. So whenever a signaling molecule binds, let's say here, for example, it can be a growth factor. So whenever it binds, this thing shall then get connected, or it shall be interconnected with another receptor tyrosine. Then they form what we call a dimer. They will come together. They work in pairs. So what happens is this. So when my signaling molecule binds here, with number one we say the dimerize. This dimerization activates the tyrosine. But this tyrosine is very special, that it is associated with enzymes called kinases. A kinase is an enzyme that phosphorylates molecules by either adding phosphates or removing them. So these tyrosine proteins here are associated with kinases. So whenever the signaling molecule binds here, we said the receptor will dimerize, then the tyrosine proteins here shall be activated. On top of that, the kinases shall also be activated. Therefore, they shall get ATP and remove phosphates and transform it to ADP. So The phosphates, these phosphates that will be removed shall be added to these tyrosine proteins here. There shall be addition of phosphates here. However, when these tyrosines are then phosphorylated, that means they become activated. We have some freely floating proteins within our cell cytoplasm, what we call relay proteins. Relay proteins. Relay proteins. So these relay proteins shall come and pick phosphates from these tyrosine proteins here. So they will be also activated, but the activation shall be a result of additional phosphates. So I repeat, signaling which binds on the cell surface membrane, causing dimerization. Dimerization activates the cytoplasmic tail. The cytoplasmic tail contains tyrosine and kinases. The kinases shall get ATP, remove phosphates, add them to the tyrosine proteins. Then the tyrosine proteins shall phosphorylate other relay proteins that shall go on triggering a cellular response. Then these receptor tyrosines are very important receptors in the cell cycle. In the cell cycle, as we shall be seeing later, they are involved in a pathways like MAPU, mitogen activated protein kinase, mitogen activated protein kinase pathway, which we shall be seeing later. Since they play a very big role in the cell cycle, any effect on them shall cause what we call cancers. Cancers. In fact, researchers found out that people that have breast cancer, their cells of the breast are associated with excess receptor tyrosine kinases, which they gave a special name as HER2 receptors. HER2 receptors. So whenever you read texts and you find this word, just know they are speaking of the receptor tyrosine kinases, but of the breast. So the having excess of these HER2 receptors means that they shall have uncontrolled cell division. So scientists developed a protein that they call Herceptin. So this Herceptin binds here. When it binds here, it prevents the activation of this cytoplasmic tail, thereby inhibiting uncontrolled cell division. Uncontrolled cell division. So having looked at the receptor tyrosine, I want us to look at what they call tyrosine associated receptors. Tyrosine associated receptors. So these tyrosine associated receptors look exactly like the receptor tyrosine kinases, but the major difference is that for them they have no tyrosine. They have no tyrosine on their intrinsic tail, as you've just seen the ultrastructure of a receptor tyrosine kinase. So these tyrosine associated receptors have no tyrosine on their intrinsic side, but they have the power to phosphorylate. They have kinases that phosphorylate. Freely floating kinases. I mean that phosphorylate freely floating tyrosine. They have kinases that phosphorylate freely floating kinases. So that is the major difference between the tyrosine associated receptors and the receptor tyrosine kinase. And these ones have no tyrosine on their intrinsic side, but they have kinases that phosphorylate freely floating tyrosine. So these tyrosine associated receptors are involved still in various pathways, like what they call the JAK-STAT pathway. The JAK-STAT pathway, the Janus kinase signal transducer activator of transcription pathway that we shall be seeing later in the regulation of the cell cycle. So after having seen the tyrosine associated kinases, next we are going to look at the receptor serine, theonine, the receptors that have serine or theonine. So these ones look like the receptor tyrosine, but the difference is that instead of having tyrosine on their cytoplasmic tail, they have serine. They have a combination of both serine and theonine proteins. A combination of both serine and theonine proteins. And they are of two types. Is what they call class 1 class. They are in fact they are of two classes. So there is what they call type 1 and type 2 and type 2. So how do they work? When a signaling molecule binds on their receptor, it will activate this type 2. The type 2, when it is activated, then it shall phosphorylate. It shall add phosphates. It shall add phosphates to the type 1. To the type 1. Then this type 1 shall then phosphorylate. Shall then phosphorylate other molecules triggering a cellular response. I repeat, when a ligand binds on the receptor of serine and theonine, shall activate the type 2, then type 2 shall activate type 1 by adding. So now we look at the last type of membrane boundary receptors, and those are the ion-gated channels. And this one, they say that they can also be called ligand-gated channels. So this is just as you hear a channel. That I'm assuming if this is a gate, this is a gate, but on it there is a receptor. So whenever the signaling, I'm assuming this is my cell membrane, So whenever a signaling molecule binds on the receptor, the gate shall open. So when the gates open, materials can enter into the cell membrane. That is it. Whenever the ligand binds on the receptor, it causes a conformational change where the gate opens. When the gate opens, materials or ions enter the cell. So that is very basic for union-gated channels, but if you really want to go into details about this, there is a video about facilitated diffusion where we discuss in details the role of ligand-gated channels in facilitated diffusion. So that is where we saw the role of ligand-gated channels, and that is where they basically play a role in facilitated diffusion. However, they can sometimes be called voltage-gated channels. So voltage means something like electricity. That if this side I have very many positives and this side I have very many negatives. Depending on what is supposed to enter or leave the cell, that is where the gate shall open. And that is it for ion-gated channels. Please consider subscribing, and thank you for watching.